Hello, in this tutorial we're going to take a look at the major motor structures within the brain by using some of the data from the Visible Human project. And in this case we're using data from the female donor. So there was a male donor, a female donor and a number of others actually. And the basic idea of the Visible Human project, for those of you who aren't familiar with it, is that the donor was effectively sectioned into very thin slices, and each slice was photographed using visible light. So this is very, very different to what we're used to seeing in terms of the grayscale images from CT and MRI. In this case, we have full colour imaging, and this does add an extra dimension and makes the images feel much more meaningful. It's well worth going and doing a bit of reading about the Visible Human project, because the techniques that they used were really, really interesting. And strictly speaking, these donors weren't sectioned. They were effectively ground down by using a special machine called a cryomacrotome. Now, as I said, I'm using the female data set from the Visible Human project, and the good thing about using the data set from the female donor is that she was cross-sectioned into 0.3mm slices as opposed to the 1mm slices that the male donor was sectioned into. So this gives us higher resolution data. So looking at the 3D rendering that I've managed to produce, essentially just by thresholding the images and removing the matrix that the body was embedded in, we can see that there's a pretty good resemblance to a human head, but there are some artifacts here, and this is essentially this artifact here has been created by me being unable to threshold out all of the gelatine that the body was embedded in order to support it during the processing. But this hasn't really affected the internal anatomy, although there is one very interesting piece of artifact that we'll discuss right at the end, which has some important clinical implications. So what I'm going to do now is just angle the head to something like this, and then we're going to slice down in the transverse plane. So I can just drag this slider here and slice down in the transverse plane and take in all of the major structures within the brain, and in fact the neck as well. But what I'm going to do is take this slowly. I'm going to go down through the transverse plane, and I'm occasionally going to bring in the coronal plane as well. We're going to identify the structures, and then I'm going to add on some 3D renderings of those structures so that we can see how these organs which are present in the sections relate to actual three-dimensional structures. So thinking about the motor pathway, we start right at the top really in terms of the motor cortex. So in our sectional view here, we're looking here at the gyri and sulci, and this is roughly where the primary motor and sensory cortices are. I'm not going to talk about specifically identifying these in a sectional view, but suffice it to say that we can make the assumption that this is roughly the area where the primary motor cortex is. And that is grey matter, of course. In fact, coming up more pinkish than grey, because we're looking here at real coloured tissue. As we make our way down through the brain, we start to note that there is an increasing quantity of white matter. So here, this is the white matter of the cerebral hemispheres coming into view here. And specifically with respect to the motor pathways, the part of the motor system that's within this white matter is known as the corona radiata. Okay? Now, the corona radiata, I like to think, it translates as the radiating crown, which is referring to all these fibres radiating out to the different areas of the cortex. So you can think about this crown of radiating fibres on your head like this. Now, I like to think of the motor pathways, and particularly the corona radiata, as being of a similar shape to my hand. So if I hold my hand up here, I like to think of the tips of the fingers as being the cell bodies of the upper motor neurons sitting within the motor cortex. And then I like to think of the fingers here as the radiating fibres of corona radiata. So this is what we're looking at here, corona radiata there. As we descend down further, what we note is that the corona radiata increasingly begins to condense. Okay, so we're getting to a point here, which we can see just about here and here, that the corona radiata is starting to condense down, to get much thicker, because obviously it has acquired many more fibres. As we descend through the brain, we will see that this corona radiata condenses down even further until we get to a region which we could think of as being at the level of the palm of the hand, where we're going to form the internal capsule. So let's look at the formation of the internal capsule. So here is the corona radiata there, and we're going to descend further. And now we can start to see how it has blended into the internal capsule. So just watch this region of white matter here, and watch how it goes from this quite chunky piece of white matter to more of a V shape in the transverse plane. So going down, down, now we have blended into the internal capsule, and we're forming a kind of a V shape here. So this is the internal capsule proper. It is V-shaped at this level. So how do we define the internal capsule, and how do we define its different parts? Well, the internal capsule is composed of 3 regions, and let's zoom in a little bit on this to give us a better view. The internal capsule is composed of 3 regions. We have a so-called posterior limb, an anterior limb, and the genu. And we can think of the internal capsule in the transverse plane as like a leg, if you like. Genu means knee, so the bend is at the genu. The anterior limb is here, and the posterior limb is there. The posterior limb is the important one from the point of view of our corticospinal pathways, because it is in the posterior limb that our upper motor neuron axons run. The anterior limb does have important motor functions, but this is more involved in communication between the cortex and the cerebellum. So most of what we're interested in is found in the posterior limb and at the genu. Now, the anterior limb is defined as that part of the internal capsule sitting between the caudate nucleus, visible here, and the lentiform nucleus. So let's look at these in a bit more detail. If I just scroll back up a little, we can see that as we descended down through the hemisphere, we saw a structure coming into view, grey matter sitting at this point just medial to the corona radiata. And as we descended further, we saw that this structure became even more prominent. So at this level here, we're looking at the head of the caudate nucleus, and the caudate nucleus is a part of the striatum. It's one of the basal ganglia. And the anterior limb of the internal capsule is sandwiched between the head of the caudate and the lentiform nucleus. What's the lentiform nucleus, I hear you ask? Well, if we scroll down a little further, we can see the lentiform nucleus a bit better. We can see it is triangular in shape, so this is the lentiform nucleus here. There's one on each side, of course. And it is composed of 2 subcomponents, the darker putamen and the lighter globus pallidus. So this lentiform nucleus here is very important with respect to the internal capsule, because anteriorly it sandwiches the anterior limb between itself and the caudate, whereas for the posterior limb is found between the lentiform and the thalamus. This here is the thalamus. Don't worry if you're struggling to visualize these in 3 dimensions, because we're going to look at them as a 3D model superimposed on this in a little while. So here is our internal capsule, sandwiched between the lentiform nucleus and the caudate, or the lentiform nucleus and the thalamus. And kind of we can think about this, we're pretty much at the level of this point here on the palm of my hand. And if you look at the palm of your hand, you can see it is kind of V-shaped, a bit like the internal capsule itself. So let's follow this further down. We're going to follow the internal capsule further down, primarily the posterior limb and the genu. But before we do that, let's bring the coronal plane in. And if I bring the coronal plane in, you can see how it marries up with the transverse plane. Look over at the green section here on the right hand side, and I'm going to zoom in on ever so slightly. When you're looking at a coronal section of the brain, you can easily recognise the internal capsule, which I'm looking at here, because it is sandwiched between the thalamus and the lentiform. Look, there's the lentiform. It's triangular here, once again with the putamen dark and the globus pallidus light. And the internal capsule is visible as a diagonal band sandwiched between the lentiform and the thalamus. And very nicely on this 3D view, you can see how this diagonal band, visible on the coronal section, is simply one portion of this entire three-dimensional internal capsule. This view reminds us that the internal capsule has depth. Okay, it's not just a 2D structure visible on a coronal section. So let's leave that coronal section in place and let's descend down in the transverse plane. So we're descending down now, and we can see the internal capsule is descending either side of the thalamus. And it's a very nice aside actually for us to just consider, why is this called the internal capsule? Well, the internal capsule is called the internal capsule because if you're dissecting the brain from lateral to medial, you note that the internal capsule forms a thick covering or a thick capsule around the thalamus. So the internal capsule is called the internal capsule because it encapsulates the thalamus. Okay, I think that's a nice little aside because I always remember being confused about why the internal capsule was called that. So let's continue to descend, and we can see this diagonal band of the internal capsule descending down in the coronal plane. And, sorry, I just needed to adjust that there. Now we're going to continue to descend down in the transverse plane. Down, down, down. And now we are at a very special level. Let's just leave the 3D view for the time being and go to the image at the top right hand side. Because where we are now is at the level of the midbrain. And the midbrain is very typically shaped because it almost looks like a face. There's a mouth here, which is the cerebral aqueduct. There's two eyes, which are the red nuclei. And there are ears, which are structures called the cerebral peduncles. Okay? So these cerebral peduncles represent the point at which the internal capsule condenses down even further to go and enter the brainstem at the level of the midbrain. On the 3D view, we can see here is the internal capsule descending down and then going into the cerebral peduncle just there. And from the cerebral peduncle, we are going to descend further into the brainstem. So let's watch us descend down through the brainstem. I'm going to zoom out so that we get the full view in the 3D window. So let's just watch this region here and see what happens as we descend down. So we're descending down now, and you can see once again beautifully the cerebral peduncles here. And that dark tissue just deep to them is the substantia nigra. Okay? So we're descending down through the midbrain, and now we're in the pons. And look, if I bring the plane of section in the coronal plane just forwards ever so slightly, we can appreciate that these motor pathways, having formed the internal capsule, then the cerebral peduncle here, then go down to enter the ventral or anterior pons. And it is from there that we enter the medulla. All right? So if I go down back very slightly, here are the pyramids of the medulla, which if we messed about enough with the settings, we would be able to show as being continuous. In fact, we can just about see it there. There are the pyramids, and they are continuous through the pons with the cerebral peduncles, which are in turn continuous with the internal capsule, which are in turn continuous with the corona radiata. So that's the motor pathway using transverse and coronal sections. Now that's all very well, But the problem is that this is a difficult pathway to appreciate. And really, what we need to be able to do is add on some three-dimensionality to this. So what I've done is I've prepared some models of the basal ganglia just on the one side, and I'm going to try to use that to show you what's going on with the fibres and their relation to those deep nuclei. So I'm just going to look at my other screen and switch on our basal ganglia model. All right. So here are the basal ganglia that I have added on as three-dimensional structures onto our visible human brain. I'm just going to take away the coronal section for the time being, and now we'll scroll up through the brain and just do a similar thing a little faster, but taking in these 3D structures. So here is the corona radiata in the deep white matter of the hemispheres. And remember, we said as we descended down, we started to see this grey matter structure here. And that grey matter structure is being rendered in 3D in this green colour, and it is the caudate nucleus. So we can see here is the caudate. All right. Now the caudate, if I scroll down, the caudate is in fact C-shaped. We can see it just there. And actually, this segmentation hasn't taken in all of the caudate. If we had the full segmentation of the caudate, we would see it going all the way around like this, following the lateral ventricle. But this is the caudate nucleus just here. And the caudate nucleus was visible up at this level just here. As we descend, what else do we see? Well, medial to the caudate at this point is the thalamus, all right? And the thalamus is a huge structure. The thalamus is a colossal structure, really, and it's formed of many subnuclei. We can see these subnuclei just here. So you can see the thalamus just there, and it's visible on the transverse section at that point. If I bring in the coronal, we'll be able to appreciate what the thalamus looks like in the coronal plane. And once again, you can see there's the thalamus, there's where it would be on this side. And I think of the thalamus as effectively being in the armpits of the ventricular system, if you like. So we've got the lateral ventricles here, the third ventricle down in the midline, and the thalami sit either side of that third ventricle under the arms of the lateral ventricles. So what else comes into the view? Well, we've also got the lentiform nucleus, remember. And the lentiform nucleus is visible here, and it has three components. Most laterally, we have the putamen. So here's the putamen visible just there, and it is quite dark in colour. Sitting medial to the putamen, we have globus pallidus, and you can see this pale region of tissue here sitting just medial to the putamen. And globus pallidus itself has two sub-components. The external segment in the blue, and in orange the internal segment. So if we take the cross-section all the way down, we can appreciate the structure of these and how they correspond to the sectional anatomy of the brain. Now, let's go back up and let's think about how these are related to the internal capsule. So I'll choose about this level here. Here is the internal capsule, remember, sandwiched between either the thalamus and the lentiform or the caudate and the lentiform. Looking at our 3D model, once again we can see that between the thalamus and the lentiform is the posterior limb, and between the caudate and the lentiform is the anterior limb. And at the tip of the point of the internal segment of globus pallidus, i.e. where this triangular lentiform nucleus points, we have the genu of the internal capsule there. So here's the genu there. Now, the genu is important, as we said, because that's where facial upper motor neuron axons run. And the posterior limb is important because this is where the rest of the upper motor neuron axons run. And in order to appreciate this, what I'm going to do now is to superimpose on this model some DTI data, some diffusion tensor imaging data showing the projections of the fibres. So let's pop that on now. So here we go. And I've colour-coded this for you. So in the, I don't quite know how to describe this colour, purpley colour, we have upper motor neuron axons which are ultimately going to supply the face and some of the upper limb, and so they're coming from the lateral motor cortex. And in red, we have upper motor neuron axons which are going to supply the lower limbs, and they're coming from the medial cortex. Okay, and you can follow you can actually see quite nicely here how they're sitting either at the genu or in the posterior limb. And if we scroll up through the slices, we can see them distributing via the corona radiata. So let's travel down now, back down to the level of the basal ganglia, and let's take a look in this space here between the lentiform and the thalamus, or between the lentiform and the caudate. And you can see very, very nicely just here the facial axons running at the genu and the lower limb axons running through the posterior limb. And if we zoom in on the transverse section, then we can see the purpley axons here for the face running near the genu, and the red axons here for the lower limb running more posteriorly. Can we show that these fibres are visible in the coronal plane? Well, yes, we should be able to do that if I can get a nice angle. Let's try to demonstrate these fibres running in the coronal plane. So here comes the coronal plane, moving in an anterior direction. And now we can see, let's just do it for these fibres for the face. We can see here is the diagonal band of the internal capsule within the visible within the coronal plane, and we can see it running through the posterior limb, sandwiched between the lentiform and the thalamus. Now you might be wondering, oh, some of these fibres seem to be doing strange things. For example, we've got fibres visible here passing through the thalamus, and there are two reasons for this. Firstly, this DTI data set is not from the visible human donor. Okay, this is from a different donor. In fact, it's from a large sample of donors. And so we're looking at an average, if you like. And of course, whenever you're doing things statistically, there is never perfect correspondence between any two individuals. And so that's one reason. And secondly, anatomy of the brain, like anatomy of any other part of the body, is variable. And so sometimes these fibres do take aberrant courses, which we wouldn't necessarily expect. But still, from a statistical point of view, most of them get where they need to get. Right, so that is our tour of the motor pathways in the basal ganglia. One final thing I want to point out to you, and that is something to do with an artifact which is present within the visible human female data set. And I'm just going to turn off our various 3D representations. And I'm probably going to use the sagittal plane as well as the others to demonstrate what I'm talking about. Now, let's go to the sagittal plane here. And let's try and find the midline as best we can. Okay, we're roughly in the midline there. And furthermore, let's go down through the transverse plane, down towards the level of foramen magnum and beyond. So foramen magnum is approximately here. Okay, we can see the cerebellum and we can see the brainstem. However, something has happened, and if I zoom in onto the sagittal section, we can get more of an appreciation of that. If we look, here's the brainstem, here's the pons, and here's the medulla. But look what happens at this point. The medulla becomes very, very narrow. And what's happening here is it is being crushed by the cerebellum. And you can appreciate that on the coronal view here. The cerebellum has actually gone down through foramen magnum. This was not pathological. This is not what killed this patient, or at least we don't think that's what killed this patient. It's documented that she died from a heart attack. However, what has happened, I think, is that during the freezing process of the body, the brain tissue expanded, because that's what happens when you freeze water, it expands. But because it had nowhere to go, since it was enclosed in a rigid box, it had no choice but to herniate down through foramen magnum. And in herniating down, this caused what you might think of as post-mortem coning, all right, where the cerebellum was pushed down through foramen magnum and crushed the medulla. So this is an artifact of the freezing process, but actually it demonstrates to us what coning would look like if you were looking at that in a real-life patient. So that's all I wanted to talk about. Thank you for listening.